All right, hello, good morning. My name is Timothy Crawford. I'm gonna get my screen shared here in just a minute, and we'll get started. Okay, let's see. Let's take a look here. Can everyone see my screen okay? There is a chat box that you can actually utilize to ask questions. And I have that up. Thank you very much, JG, for the feedback there. So let's get started. I'm going to try and cover a lot of content here in the presentation and then hopefully have some time for a demo at the end of this initial seminar. And I know this topic has clustering algorithms as a focus, but we found that I think a lot of people joining these seminars really need to understand the entire workflow of employing these algorithms. And so I'm going to actually go through more than just the clustering algorithms. I'm going to talk about dimensionality reduction techniques as well, and how we can merge those tools together in a workflow that will allow you to find complex populations in high parameter data and determine how they change across your experiment. So I'm going to get started by going and through a presentation, and then I'll demo the functionality at the end for you. Now, of course, basic goals for any high parameter analysis are going to be defining your populations in some way, and you could do that with manual gating, or you could use one of these automated clustering approaches that we'll introduce you to. And then visualize the phenotypes, and visualization can be done in a univariate plot with histograms, bivariate plots showing 2 parameters at a time. You can overlay the clusters into a 2-dimensional embedding in lower dimensions. So these are T-SNE plots here that I'm showing. Or you can actually enumerate the median fluorescence intensities of each of the channels in your panel across all of the clusters to get a phenotypic property set of the phenotypic expression pattern. And ultimately, we want to understand how each of these cluster populations that are defined, or manually gated populations that you know about, change between different conditions. And here in this T-SNE plot, I've broken up into a stimulated and an unstimulated condition. And you can see that the stimulated cells have different locations within this T-SNE continent structure that I've produced, indicating that upon stimulation, these cells change, they go somewhere else because the phenotype changes, effector functions are upregulated, and they move visually within this dimensionally reduced data space to a different location. And that's what we're trying to evaluate. How do these populations change? Do they go up? Do they stay the same? Or do they go down across your experiment? So, of course, you want to define your populations manually. I think everybody has an idea of where they want to go with the panel when they're doing high parameter cytometry, and you have a hypothesis and specific scientific aims. So certainly gate down to what you know, define your different memory subsets, find your Tregs, your gamma deltas, and all the other subsets that you're interested in. But the challenge for gating comes downstream of those major lineage markers, where you have a bunch of effector functions, exhaustion markers, things that are changing as part of the immune response. And you have so many of these that it's really hard to get any type of combinatorial phenotype. You can gate them one at a time and do Boolean gates, which is complex combination gating, where you take each of the individual markers and you look at all potential combinations. But if you have 8 markers downstream of a lineage or a subset that you've defined, that's going to create, each Boolean combination of 8 markers is going to create 256 different gates. So just 4 different subsets, I'd end up with over 1,000 gates by Booleaning these together. And if you actually start looking at all the different bivariate plot combinations, as soon as you get up into the 20+ color range, there's too many of them to even evaluate by eye. So we have to come up with some sort of alternative approach for actually gating these complex combinatorial expression patterns that we see in the immune system. The other downside is that you only gate for what you know. So if you have a disease state that you're investigating and you don't know what kind of populations are going to be there, then how can you identify them with a hierarchical manual gating approach? So over the past 10, 12 years, there have been something on the order of 40 different unsupervised clustering approaches that have been published and produced by the community. And a lot of these have started gaining traction in recent years because they become available in commercially developed applications like FlowJo. And the three that I'm going to push you towards are the ones that we have decided to incorporate into FlowJo, and they are Phenograph, XShift, and FlowSum. You can see in my word cloud here. And there's different methodologies here and some great reviews going through the different approaches that are out there, but all of them do the same thing, which is create populations. You get populations of events that have been classified by the math and the algorithm in an unbiased and objective way to give you a summary of the distinct combinatorial states. And I think all of us can appreciate looking at these histogram traces of the different cluster populations that I produced here, that sometimes the histograms and the marker is just a completely bimodal distribution. It's either on or it's off, and it marks a major lineage or a specific subset of cells that pop right out with the clustering. But then in other cases, like CCR7 or CD95, you have a continuously variable expression pattern, and there are intermediate levels of expression. That is, there are populations there that are transitioning and upregulating and downregulating the marker to do a different function, whether that's trafficking, cell death, effector functions, exhaustion, whatever. But it's not going to be as clearly bimodal and distinct. So it's not easy to just gate what's positive and gate what's negative. If I do that through CCR7, I'm cutting through these transitional intermediate states with a hardline gate and not capturing the complexity of that marker. And I think that really the clustering algorithms do a good job of giving you those intermediate phenotypes that may be the transitional states in an immune response. And our job, once we have those populations, if we didn't define them with a hierarchical gating structure, is to visualize and call the phenotypes. As I said, in this case, I'm doing a heat map of all the median fluorescence intensities for each marker, and then understand how the populations change across the experiment, whether that be unstimulated versus stimulated cells, sick versus healthy, knockout versus wild type, whatever conditions you can define, we can make that comparison across them. So these clustering algorithms, they create populations, and then the dimensionally reduced data space gives you some way to view all the data in its entirety, even though it's a highly complex panel. T-SNE plots have been the classic over the years. They were the first plot type to be coded into FlowJo, and it's a native T-SNE function with all the various options to do that particular calculation. And but I think I need to take a step back here and tell you how I even get to this point and why we might use T-SNE versus another dimensionality reduction approach and what the different options are here. And so let's take a look at just the workflow we might employ both dimensionality reduction and clustering tools within to get that answer. What is in our system and how do those populations change? Well, first off, we're going to clean up our samples, right? You aren't just going to dimensionally reduce or cluster on your raw data file with debris and dead cells in it. So you can gate just like you're familiar with in a hierarchical way, drop those gates onto a group so they all the samples get gated. In this case, I've actually used an automated plugin approach called Flow AI to actually scan through the original file right at the root level of the file and cut out any bad events that were outside of certain thresholds. So the autofluorescent events, clogged areas that show a decrease in scatter, they'll get removed in this automated approach. And then I'm gating away my singlets and my dead cells and coming up with just a nice population of live lymphocytes. So once you have clean populations, you can actually merge those samples together using a process called concatenate. And during that concatenation step, we can choose to make the number of events from each sample going into the concatenated file equivalent by stating we only want to take X number of events from each sample. Because as you collect different numbers of different samples, you're going to have different numbers of events generally. And even when you clean up, they're going to be different, right? Even if you did a stopping gate and took the same total number of events, once you gate down to a clean population, you're going to have different starting points. And it makes sense to just kind of balance the equation from one side of the experiment to the other and have the same number of cells from each sample, patient, or animal. You don't have to. You can always just leave the raw cells and deal with the frequencies there, but it makes a lot of sense to balance the equation out. During the concatenation step, we can create parameters and actually give each one of these sample files a unique value to organize them into a grid that allows us to pull them back apart. I could gate on a single sample or gate apart on the X, comparing tissues, gate apart on the Y, comparing time points. But the purpose of this concatenation is that we want to make a common data space with dimensionality reduction and a common set of clusters with a clustering algorithm that actually incorporates and considers during the algorithm all of the data, all of the distributions from all of the patients and conditions. If you actually do a dimensionality reduction on each individual sample, you'd get a completely different continent structure, a completely different map out of that. And it's no longer going to be comparable across samples. If you take all the data, merge it together, and do one dimensionality reductions approach at the same time, then you get a common data space. You can cluster on that data all together and get a common set of clusters. And then we can view the clusters, determine the phenotypes, and still go back and pull apart the concatenated file. Now I'm gating across the tissue parameter that I created to view either see what the frequency of clusters are across of any given cluster across the conditions, or visualize in the common dimensionally reduced data space the events that are coming from, in this case, the different tissue compartments of this animal. And when you do that, you'll actually see that there are regions of density here, events that are enriched in certain conditions and depleted in others, right? And that's essentially what you're looking for. And so as long as you get the concatenation step down, which method you use to display those populations or that data within a dimensionally reduced embedding, and which method you use to subset into cluster populations, it is really just which tool is best for your use in terms of how fast it runs, how many populations it'll make, whether you have control over visualizations as part of that process and such. So that's the workflow that I'm promoting, because I think it actually works to answer those questions of what's in my system and how do those populations change across the conditions. Now remember that dimensionality reduction is just a visualization, and all you get out of a dimensionality reduction algorithm is two new parameters, an X and a Y. And these parameters redistribute the events into a two-dimensional space, creating this continent-like structure with different islands. And each of the islands is going to represent a different phenotype, right? That's the principle, is it separates out the data, it sorts it out into two-dimensional space based on the high parameter expression patterns. So if you overlay any gating that you create for populations that you know, such as I've done here with lineage markers for CD4, 8, 56, 14, and 19, just gating on the major lineages, if you overlay those populations onto your T-SNE plot containing all of the events, you will see that events with a similar multidimensional expression pattern group together within this dimensionally reduced data space, as they should, because that is the principle of what we're trying to preserve, is that high dimensional data represent, the structure of the high dimensional data represented in a two-dimensional view. Now, one thing you can do once you have a dimensionally reduced data space to orient yourself is not just overlay known gated populations, but turn on a third parameter color map option in the FlowGeo graph window and pick another marker, and that will heat map across your continent the expression of that particular marker as higher redshifted regions. And so you can easily see that these colors correspond to the locations of my overlaid populations, because I was gating on those markers, and that's where they are expressed. What you actually decide to dimensionally reduce depends on the question you're going to be asking and what your panel is targeting, what reagents you're actually using, what antigens you're focusing on. In this case, I've got an example, which is a 25 color panel that is run on whole blood, and it's got all the major lineage markers in it. So if I just gate for CD45 here and then do T-SNE and overlay my lineages, you can see that granulocytes, which represent 70% of that whole blood sample, actually dominate this dimensionally reduced embedding, this visualization. And if the question I'm asking in the immune system has nothing to do with granulocytes, well, then I'm just wasting space here and wasting resolution that I could get by removing the grans, and I'm just gating away the CD15s, keeping everything else. And now you see all these different islands which were on the periphery get promoted into the center and spread out, and you see more structure. So as you progressively remove lineages with sequential, in this case, negative gating for each one, you get more resolution and higher structure available to you for the cells that are left in that particular subset. So if my focus is I want to look at CD8s and look at memory and exhaustion in CD8s, then maybe I'm not going to do the dimensionality reduction or clustering at a higher level. I'm going to go right down to my lineage and then subset within that set of events with a clustering algorithm and display it with dimensionality reduction in this two-dimensional view. So what kind of options do we have for dimensionality reduction? The classic T-SNE is probably what people are most familiar with. T-SNE does a great job of flattening the data, and it does a really good job of not overlapping different populations that have different phenotypic expression patterns on top of each other. UMAP is a newer method that has been introduced since about 2018 and gained a lot of popularity in the single cell sequencing world because it scales better with the number of dimensions. You can actually take hundreds or even thousands of dimensions into UMAP directly instead of going through a stepwise process to get down to 2. And the UMAP does a better job of preserving the global structure relationships between lineages, in that you see T-SNE start to break up its lineage compartments. As you get more complexity, you'll see these islands that are related to each other but may not be in the same on the same side of the continent or even within the same island. And UMAP does a better job of preserving those and not having the fracturing, but you do get this effect of having multiple populations sometimes layered on top of each other. TriMap is one of the newest options out there. We've implemented it as a standalone plugin, as we have with UMAP, so neither of these require our setup. And it does a principal component analysis first and then modifies the PCA with a selected triplet from the data. And then embed-sum is essentially just a view in single cell world and a single cell dot plot of the grid layout of a flow-sum clustering output. So this is a 2D visualization specific to flow-sum. If you run flow-sum as the clustering algorithm, you can then run embed-sum and get this view. And it is similar to PCA. It's showing the self-organizing map and separating those events, but it shows the actual single cells and their positions within that map. So by far and above, most used are T-SNE and UMAP on flow data, and in single cell multi-omics or single cell sequencing, UMAP is the clear winner in customer use right now. Both of these give you valid representations of your data, but they do have the difference here, as I'm going to say again. I mentioned it, but that T-SNE does a better job of flattening all the data, so you don't have anything piled up on any different populations piled on top of each other in the z-axis, and it preserves more local structure. So events within this island are going to be similar to each other, but you cannot make any inference that the position or relationship between islands has any meaning here, because you will get this fracturing and will find, your CD8s will start having islands all over the place. And so you can't say that because this is closer to the 8s than it is to the Bs, that it's more closely related there. You can't make that inference. UMAP, however, it keeps the global structure preserved, and it does not have the same type of fracturing of lineage islands into multiple islands in different locations here. And so you can actually make some inference here that if the cells are closer to each other, that they're more closely related. So that's just global versus local structure. And the way these process is similar in that they're iteratively processed in a way that gives you this ultimate continent projection in the end. And so what T-SNE does is it converts the high dimensional data into the Gaussian probability distribution, and then it seeds the local data, initializes with seeding the local or the low dimensional data, converts that into a probability distribution, and then compares the two probability distributions for how close they are to each other. And they use this method called relative entropy, or the Kullback-Leibler divergence. And that is one round of the calculation, one iteration. And with each iteration, each cell looks around and selects its nearest neighbors, and it pushes on those. If they're dissimilar, they get pushed further apart. It pulls on them if they're similar, and they get pulled closer together. So with each iteration, these events move slightly, then it converts and compares. Moves slightly, converts and compares, right? And after it does that 1,000 times, it gives you this final resolution or embedding with an X and Y parameter redistributing events. UMAP does a similar thing, but it does it using fuzzy simplical complexes instead of Gaussian probability distributions. And it converts and compares those fuzzy simplical complex manifolds over the data set using a method called cross entropy, but it's still an iterative process. And it seems like it's really in the initialization of the process where this global structure versus more local structure gets set. But they're both very relevant visualizations that confer nice, graphical imagery to an audience. And if you break it down, you can compare the shifts and differences between different conditions visually in a two-dimensional plot, even though you have a 20+ color experiment, which you wouldn't be able to show all those plots in 2 dimensions at a time. So just to reiterate on the concatenate step, the only way to get a common dimensionally reduced data space to compare different conditions or sample files here is to concatenate your files together first. And during that concatenation step, you have the opportunity to add what are called keyword labels, indicating what bin or experimental condition these samples are in or from, or were processed from, right? So in this case, I've got 4 different time points and 4 different tissues within each time point. And so I may label my keywords, and then during the concatenation step, you can choose to create additional parameters based on those keyword labels. And that gives me the organization here, 1, 2, 3, 4, 1, 2, 3, 4. And now I can create the T/E, gate apart, knowing that I normalize the number of events. So I've got 25% of the cells coming from each tissue to each time point. And then breaking that out, I've shown these plots before, but you can easily see regional expression in the different tissues that correspond to the different cell types. Lymph node has more Bs, 4s, and 8s. Spleen has some special B cells. Liver has a bunch of innate cells and CD8s. Blood has, 4s, 8s, NKs, and monocytes, and some Bs, but they're slightly different in phenotype than the others. So once you know what the map looks like, based on your investigation with overlays and color maps, you can make those determinations of what these regions represent and easily visually see that they are contributing different populations to the system here. Just a little more detail in case everybody's already confused with the keyword situation. Keywords can be created as metadata labels within your workspace in columns. And you do that by going to your workspace tab, adding a keyword, giving it a name. And I always put an asterisk in front of my names. That makes them unique. It also gives me a way to find them in a list of all the different header information that keywords join. So I can order my samples in any way that I want by doing an order keyword. This is my time points, so 4 1s, 4 2s, 4 3s, 4 4s to organize them along my time point axis. And these are my tissues, 1 2 3 4, 1 2 3 4, 1 2 3 4, and so on. Right, so that's just organizing the grid so that when you concatenate, you can actually do something with it. And you can make that comparison that you want depending on your experiment and the study that you're trying to, the hypotheses that you're trying to test. So what can you do with this dimensionally reduced data space? Quite a bit, actually. I've shown you can heat map across the data space any other marker. So this is checking a color axis box here in the graph window and choosing another parameter to see where it is distributed, where its expression pattern is in this continent structure. You can also make what I call a query gate, which is the easiest way to explore this data space in terms of what's the phenotypes underlying the different regions. Because a third color can show you one marker at a time, The query gate, you can set this up as an overlay in your layout editor, just dragging the query gated population on top of the root file, displaying T/E. And then if you right click on this, you can show multigraph histograms. And that's what I'm doing here, showing the magenta trace is what the expression pattern is of the query gated region, and the gray trace is all of the cells in the system. So it's easy to phenotype just by moving the query gate to a region that you're interested in and then looking at the expression pattern. Now, if you take that query gate population and you actually drop your experimental condition gates downstream of the query, well, that'll give you the frequency of those cells, right, where they came from. And if this is my starting point where I know I put 25% of the cells from each of the different compartments into my system, then my query gated population shows different frequencies. In this case, the spleen showing 70% of these cells here came from the spleen, only 1% from the lymph node, 10 from the blood and 20 from the liver. Well, that's a huge difference than having 25% across the board. If everything were 25%, I would say, well, this population, whatever its phenotype is, isn't changing across my conditions. In this case, I can say, oh, this phenotype, which are CD95 positive B cells, is actually expressed at high levels in the spleen relative and virtually absent from the lymph node, right? And make that observation and that conclusion. You can see it here in a different way where you drop the query gate down onto the T/E plot and you're just gating for that node. But I like to look at it this way because now my query gate, anywhere I move it, I get the frequency across that where those cells came from, what the different samples were that they're derived from. Here, it's kind of looking at it a little bit differently, and it's not the way I like to think of things. So yeah, you can do a lot with just a query gate. And of course, if you run a clustering algorithm, you can overlay those clusters and view those histograms as well. Now, what you really want out of the clusters, as I mentioned before, the MFIs, the expression pattern, not just a visual expression. This is hard to read when you get 32 clusters here. But this is really easy to read, right? It's just a heat map. And in this case, I did this one in Excel using the standard table editor export from FlowJo. And the method to make this table specifically out of the table editor, because the table editor is designed really only to iterate across samples, not within a sample across populations. So if I want this orientation, this nice grid, what I do is export the cluster populations as individual FCS files. Then I load them back into my workspace and I group them together in a group. I would call this just flow sum clusters. Then I add statistics, the median fluorescence intensities for all my compensated fluorescent parameters, and count statistics, so I know how big the population is when on my exported population. And then I even drop the experimental condition gates onto the group. So you can create the statistics on a single sample, drag them all to the group of flow sum cluster populations, and then enumerate those stats across the group in my table editor, and this gives me a nice grid. So now going into Excel, I can just heat map each column from high to low, red to blue, and immediately see what clusters express what given marker, apply that to each column. And then over here on the condition gates, I'm actually applying a different color palette across the row within the group of gating here. And so this is my time course, this is my compartments. And you can easily see just with the color coding without, I hid the numbers over here, I left them over here. You can see that some populations start low, they go up and then they go down. This is a time course of response of a treatment. So that I'm expecting things to change. Some populations start out high and then they're gone, right? Those cells changed into something else. You can tell where they came from. And all the information is here, right? And accessible. And it's just a table export with a bunch of median fluorescence intensities. If you know how to export, load into a group, add your stats, and then enumerate a table, you can go and heat map in Excel here, that table export, and make a nice little visual for your group and communicate what's going on in your system. There are actual different clustering options, and some of them, which one you use is going to depend on your data. It's going to depend on how many events you're trying to cluster and whether you want some extra visuals out of it, really. The three options that we are offering right now are XShift, which is a standalone plugin that runs in Java. You don't need R for this. Phenograph, where we actually have the newest version runs in Python and is also standalone, but the older versions are in R, and I actually have observed that the R versions run faster. So when I've actually kept the R-based plugin in my build on Flojo, and I run that because I'm trying to run hundreds of thousands or millions of events, and the newer Python one just doesn't run that fast. So I'm using the R-based version of Phenograph here, which I really like, and then FlowSum is the third one. This is Sophie Van Gassen's FlowSum approach, and this is an R-based plugin. You do need to have R installed and set up to run. But you can see the clustering overlays here that some of the populations are present in some methods and others don't cut up the area. Every one of these uses slightly different mathematical approach to subsetting or classifying your events into bins, into clustering groups that actually have shared phenotypic properties. And so I'm just going to go through each one. I'll talk about XShift first. It works really with K nearest neighbor density estimation. I've got the paper reference right down here if anybody's interested in details. And to me, the big pro here is that it's relatively fast. Like it'll classify quite a number of events in a reasonable period of time, 30 seconds, couple minutes. It doesn't require you to input the number of clusters. It does this thing called an elbow plot where it kind of tests varying numbers of clusters and looks for where the variance stops decreasing significantly, and it picks that cluster number. And so it's trying to find some, not over cluster, but not under cluster. And so you just press the button, go, it's going to find whatever it finds. It is, technically optimized to find small or rare populations. And it does not require you to set up R. It's running as a standalone program from Nikolai here. And the same thing that is good, where you don't have to specify the number of clusters, can also be a deficiency. You can't specify the number of clusters. So if you want to get out a specific number of populations, you can't do that with XShift. And there's no real default visualizations. The visualizations I'm showing you are coming from my Cluster Explorer tool, which will create these overlays. This is a principal line plot that you can actually gate within, and it's showing each line is representing a different population here, and it's showing the expression pattern similar to the heat map, but in a different visual. And then this is a heat map that I can get out, which has all the underlying MFIs. And in this case, I've actually added additional columns, which are the gates for my different conditions. And so these are frequencies across of each cluster across my different conditions represented in a different color palette. So these are visualizations that we can offer you through the Cluster Explorer plugin. Phenograph is my personal favorite in terms of what it actually produces. I really like Phenograph clusters. I like the way it cuts. I like that it finds unique populations within lineages. It has a tendency to make more populations or over cluster compared to XShift. And it uses a math approach that has actually become really, really hot in the single cell sequencing and multi-omics world as well, where it uses this nearest neighbor graph construction and then partitions that graph into these communities based on Louvain community detection. And it does automatically determine the number of clusters. Again, has a tendency to overcluster. It is fairly fast, with the caveats that I've already mentioned, that I prefer to use the R-based version 2.4 over the version 3, because it seems to run faster on higher numbers of events. So I say, yes, it's fast enough to scale without downsampling. That doesn't mean it's not significantly slower than the other two methods I'm mentioning here. And again, you don't get default visualizations. You can't specify the number of cluster, although changing the K number can kind of have an effect on whether you get more or less. K is the number of nearest neighbors considered during the calculation. So, read the paper before you use it, but there are a number of papers now referencing Phenograph as a tool, and a lot of them are really biologically relevant. That means it's not just like we created this tool and published it in a math journal. We utilize this tool to do a really, really interesting study and got a cell paper out of it, right? So Phenograph is my favorite personally with respect to what it produces in terms of the cluster phenotypes. It is the slowest of the three options, unfortunately, at this point. FlowSum, probably what everybody already knows, right? FlowSum has been out for a long period of time. It gained a lot of notoriety initially because of these visualizations that come out of it. And it uses a self-organizing map and what's called k-nearest neighbors to generate these cluster populations. So it actually over clusters. It creates 100 by default mini clusters. And then the user, you have to specify how many meta cluster populations to generate. So each of the larger colors, these groups of little clusters, are actually going to be a meta cluster population that's returned back to the FlowJo workspace. It is extremely fast, and that is absolutely great, because if you just want to cluster something and you have millions of events, FlowSum will be your go-to, because it can, a couple minutes, classify a couple million events without a problem, in a way that would take Phenograph hours or overnight, potentially. I have a tendency not to test that many events up in that millions of events in Phenograph. So if you want to cluster a lot of data, FlowSum's good. If you want to have a visual like this minimal spanning tree with radial star plots here, it's each piece of the pie represents a different antigen expression. You can see that some are bigger than others. If you zoom in here, you can interpret like what the phenotype is of each of these different populations that have come out. It also comes out with a nice visual of a tree with a heat map with some dendrograms. The actual MFIs are produced as a table. So it has these additional files that come out of the pipeline in R. And another big benefit of FlowSum is that you can actually apply new data to an existing map. So when you run FlowSum the first time, it'll create a FlowSum map. And then when you go to a different population or a different sample, you can actually classify that new data that you point to into the existing map by choosing apply to map and choosing the last run. And that will give you essentially the same clustering effect, but if the sample is different, you'll have different frequencies in these different cluster populations, and the node sizes will change. You can compare them. And that's something that's unique to FlowSum and is not, we don't have an ability to do that with the other two methods. Now, I don't like FlowSum because it does not have a tendency to overcluster. You can force it to overcluster by just increasing the number of metaclusters, but I'm not always sold on where it decides to, like, if I increase the number of clusters, it's not necessarily going to chop up the B cell population here. It might just make a bunch of other small clusters in the complex area of the NK cell subsets. And why, you know, Phenograph will do a good job of subsetting certain lineages and other methodologies will not. Well, it's all in the data and how it's distributed and the math underlying where the cut points end up being, right? Where the decision is made to include this event into an adjacent cluster and not the same cluster. So I can't go down into the weeds that far for you and give you like the answer here, but I can say that I like Phenograph, even though it's slow in terms of what it produces. I like FlowSum because of the visualizations and its speed. And I like XShift because you can just download the jar. You don't have to set anything else up, and it just works on your data. And so right now I'm still at the process of like making a decision if I were to move forward with just one, which one I would use. And it would depend on how much data I have to cluster, how complex the data is, and whether you know, I have the heart and the time to actually compare these different methods to each other, which would be kind of the ideal situation as you run all three. So, you know, if I run all three methods of dimensionality reduction and three methods of clustering, how do I actually work with that data and use it? Well, we've created this Cluster Explorer plugin, which gives you this interactive ability to select different populations in the line plot, in the heat map, show 2D plots of all the various dimensionally reduced embeddings that you've created. So I've got a t-SNE, a UMAP, and a trimap here. We can create additional plots, little 2D plots here to show bivariate expression patterns of any given number of markers, show the cluster sizes, and this is all interconnected. So I can actually use gating here to identify a subset of clusters. And when I you know, highlight, when I gate on this plot, it limits what I'm seeing in the other plots. There's actually the ability to change cluster sets here. So I can look at Phenograph clusters, then shift to XShift clusters, and shift to UMAP clusters. We can plot in the heat map you have underlying data that could be means or medians, scale values or channel values, and then there's an options bar button here that allows you to just copy the content, and you could copy this table right out to Excel. You can copy any of these images out. And then finally, over here in the heat map, I can actually add additional nodes as columns. And in this case, I've chosen the stim condition gates. This is a unstim versus stimulated sample. And now I'm looking at columns here that tell me, well, how did each of these clusters change across my stimulation conditions in my experiment? Finally, we've implemented this ability to compare cluster sets with a contingency table and look at overlap of individual events, F measures, or Jacquard indices. And this is even showing matching with the Hungarian algorithm. So if you want to compare, well, do my clusters match from this method to this method? There's a little button here that you can compare two different cluster sets for their overlap and which populations are they capture very robustly, both of them, and which ones they don't capture or differ on. So now what, right? What do you do with a cluster population? You have no gating tree. You can't clean it up. This is an old Cluster Explorer UI here. So the question becomes, when you have clustering, right, how do you sort a population with a clustering, identified with a clustering algorithm? And we've kind of come up with a solution there too, called Hyperfinder. And Hyperfinder is kind of a merging of math between two different methodologies that were published about 2 years ago, GateFinder and Hypergate. So there were two publications here, and we took both of those and kind of created one Hyperfinder that you can choose to be more GateFinder-like or Hypergate-like. And what it does is it goes through and it builds a biaxial gating sequence and then optimizes the boundaries to maximize the F1 score. And that's the F measure score, which is the harmonic mean between precision and recall. All it means, F1 score, you can think if you gated on a population and I gated on a population, how much overlap is there between our different gates? Meaning, do we have 100% of the cells in your gate are also in my gate? Is it 80%? Is it 20%? So the F1 score is a fractional percentage. You see a 0.8, it means 80% of the events in the original population, which is now our cluster, were found with the gating hierarchy from Hyperfinder. So Hyperfinder splits the data, this would be the clustering events from a particular cluster that you decide you want to sort. And it splits the data into a training set and a validation set, and then it builds a series of biaxial gates. You specify how many gates deep your sorter can go. And then it starts to optimize the boundaries. The blue are what it's testing right now. The gray is what it started with. So it's like making them smaller over time. And each time it optimizes a boundary, it reports back out the F1 score. It's trying to optimize getting the most events from the original cluster into the final gated population here. Then it goes through at the end after finding optimal gates, and it says, well, can I remove any of these gates and still get a good score? So can I make the gating as short as possible? And it will do that and then eventually report out to you the final gating path, which will be returned back as gates within FlowJo. And the message here, it's actually the precision, the recall, the precision, the total F measure. So this is 95% of the events from my cluster ended up getting captured just with 4 gates here in this sequence. And it took 34 seconds to run. So I can close that. This is the actual graphical user interface. You can pick which parameters you're going to have available to you on your sorter to sort the population because, of course, intracellular things aren't going to be there. Click OK. This is the number of gates that you could use on your sorter. The F measure beta, you can actually tweak this to be geared more towards purity versus yield by changing that F measure beta number. And it's just a really cool approach that if you don't know how to get to your population, you can run this and you'll know what sequence of parameters could be utilized to purify that population for functional and downstream single cell omic studies. And you know, we even have a export to Diva function now, although it really works with data coming from your sorter. And so you'd want to run your sample, a little bit of your sample on the sorter and export your sample experiment from Diva here, but then you can actually derive your Hyperfinder gates, put them onto your sorter sample, and then export them right back to the experiment. And you can go back to your sorter and open it up, and you will have the hierarchy of gates that came out of Hyperfinder. But it's totally possible now to use this workflow to create populations using dimensionality reduction and clustering, find the population of interest. We're going to clean it up for the outliers here, run Hyperfinder on it, and then go back to the sorter and actually purify those events. You can also use Hyperfinder just to see if there's an alternative path to getting the events that you currently sort, right? You could maybe drop a few parameters or get your sort to a higher purity or yield by considering gating on bivariate plots that you would have never considered in your own manual approach, right? We have a tendency to always do the same type of cleanups, start with scatter plots and such, and Hyperfinder is not going to say anything like, I need to gate this first, because that's just the way I've always done it. It's going to say, well, what's the best parameters to use to get to that population as quickly as possible with the highest purity or yield as possible, right? So really kind of nice way to unbias your sort approach. Now, when we have sorted populations, we can actually drop them onto a gene chip now, and we can go and isolate those single cells into hexagonal wells, drop a capture bead, lyse the cell, and capture all the RNAs from the cell. And that can be sequenced and give us expression information at the RNA level that's similar to what we get in flow cytometry at the protein level. But we can also stain those cells before the capture here with antibodies that are sequence tagged, which means that we actually have the ability to create a library and sequence the proteins on the surface. So now we have RNA and protein measurements, and we can run through a similar workflow with dimensionality reduction. In this case, I just overlaid, it's not actually clustering, but there's clustering to create populations, and then do differential expression between those populations and come up with what are the gene sets or feature sets that are differentially expressed. The full workflow there is sort your population. Well, I'm sorry, generate a sorting hierarchy with Hyperfinder if it's a computationally derived population. Stain with fluorescent antibodies so that you can sort, and sequence tagged antibodies so you can get protein expression out of your sequencing experiment. You can even multiplex these using a multiplexing kit. We can do 6-way sorting now on the S6, which is pretty awesome. And then drop those onto a gene chip. We do some QA. We capture and lyse the cells, capture the RNA and protein, some PCR steps, sequence it, some bioinformatic processes, and you get an expression matrix. And our expression matrices, at least through this pipeline, are all locked down. And it's not locked down, it's the pipeline to get the bioinformatics to get the sequencing out into a usable expression matrix we have locked down and sorted out for you. And we have a service that we provide. So it's just point and click. Once you get those expression matrices, that is the single cell RNA and protein counts, just like an FCS file, we're going to be looking at data in a matrix, and we can do dimensionality reduction, clustering, and differential expression here. And it becomes similar to flow, but we have many more parameters, hundreds of parameters in a targeted assay, thousands of parameters in another, in a whole transcriptome assay. In this case, I think 32 of these parameters are protein and 399 are RNA. Well, to go with 431 parameters down to 2 T-SNE parameters, we can't go directly to T-SNE with the raw data. What we do there is go through principal component analysis to get 15 or 25 principal components, and then take the principal components and feed them into T-SNE to get down to 2 T-SNE parameters to make the map. I mentioned UMAP can go directly there, right? Where I can just feed in all 431 parameters and get a nice map, right? These are obviously different, but which one do you use? It's up to you. And maybe it's not enough to just show everything in one plot. Maybe I need to zoom in on a subset. So let's take the magenta and purple NK sorted subsets and use TriMap to spread them out. Now I've got actually a trajectory that actually reflects the differentiation of those cells, with effector functions and cytotoxicity being highest out here, and more naive common progenitor-like cells being out here. And so we can cluster on top of that. In this case, I'm using Phenograph, feeding in all the parameters, and then do differential expression across those different clusters and find the unique RNAs and proteins that confer an actual feature set that is a unique expression pattern for that particular population within your system. And so now, I mean, I'm casting a wide net here, but I get a nice, I mean, I've got more markers defining these 8 clusters than I can even fit in a multicolor flow cytometry panel. And the only way to do that is to go into single cell omics. The methodology is there, and it's just amazing to me that you can actually do this, because a couple of years ago, it wasn't even possible. So, you know, I'm not going to go through the summary. I'm just going to jump out of here and say that, you know, thank you guys for your time. I'm going to do a quick demo, and then we'll have to close it off. I realize I've been talking now for about 50 minutes, so a little longer of a presentation that I would than I was intending, but I hope it's helpful for you. If you have any questions, please type them into chat, or I think you can unmute yourself and actually ask them. And I'm going to just do a quick demo of the workflow here. Okay, so we'll hop out of the presentation, and I'll just take 10 minutes to do this for you with an example. Let's just get out here. Oh, come on, baby. There we go. Okay, so let me spin up FlowJo real quick, and I'll grab a workspace with a 28 color panel example with just a stim versus no stim condition, and we can run through the tools. Okay, so here's my workspace. I'm actually going to, well, let's just do it from scratch. So grab myself some data here. There it is. Okay. Going to grab, yeah, I'm going to use a template. So here's a workspace template. Templates are something that you can use to preserve all the gating and the layouts and the tables and the organization that you've created, and even the scaling on a set of parameters by saving as a template. So file, save as, template would give you a WSPT file. When you open that up, you won't have any data, because that's what a template is, right? Organization, structure without data. And then you can bring in data files. In this case, I'm going to grab the 2 files from OMIP 060 and bring them in, drag and drop a stimulated and an unstimulated condition here. These come in without any actual compensation, but the template also keeps your compensation matrices. So I'll just drag that on. And now we have compensated data, and the gates that we're living on the group here. Once the compensated parameters exist, then it goes ahead and gates. So I'm going to choose, let's just say, a common upstream gate for lymphocytes, and we'll take that and dimensionally reduce it. So you gate down to some lineage, and then the next step would be to merge those together. But before I merge, I want to make sure that I have my different stim conditions labeled here. And I've already created in this template a stim condition keyword column, so it exists. But just to show you how it works, keywords under the workspace tab, you can add a keyword and you can say, this is my experimental condition, right? Just a name that'll become a column here, and you can give these columns integer values. You can actually create keyword value series in a repetitive way using this keyword button here. So 1s and 2s, and I actually want to do it the opposite way. I'm going to do a 2 for the PMA stim and a 1 for the other stim, or for the unstimulated. And these columns can be controlled by edit columns. I'm right clicking on the column header here. I can also sort by a column header. So I'm just going to sort this one ascending. So I've got 1 and 2. I'll go ahead and do this too. Edit columns, goodbye, my experimental condition column. I'm removing it so it's no longer shown. So this will be the order of my stim condition keyword. We don't want every single cell here in my lymphocyte population, because it wouldn't be balanced. You would have lymphocytes that are 138 versus 146. Right click gives you a drop-down menu. You can choose to select equivalent nodes here. Then I'm going to right click again and choose to export or concatenate. This is going to be a concatenate step, and I'm sorry if I go fast here for time purposes. There's a lot of options in here. What you really need to know is that if you stay on the export tab, you create 1 file per selected population. If you concatenate, then you merge those populations together. I'm going to check a box here under include events to include no more than, and I'm going to go down to just like 25,000, because I don't want the TSNE to run too long. in a demonstration example. So I'm just going to stay with a low number of events here. Under advanced options, we can choose a unique name. So I'm going to call this lympho example, just to give a unique name. This is the location of where my data will be saved when I export. Generally, you're going to use all uncompensated parameters that will give you the matrix plus the data. But you can use a custom set of parameters, or you can go all compensated parameters, but then you're going to only write compensated data to the file, and you will not have a matrix or any raw original data there. Now, the key point that I want to make in this user interface is include events, right? Include no more than that normalizes across the samples to a select amount of number of events per sample. And then additional parameters. This is where I can choose the keyword that I created here, stim, with the values to organize my samples in a way that is meaningful to me so I can pull them back apart and make the comparison across the conditions. So choosing the sample here pops up this additional parameters window. These are all of the possible choices. These are keyword headers in the FCS file. And my asterisks are right down below the dollar signs. Dollar signs are all required information in the FCS standard. There's a bunch of other information that's alphabetized below, but I like to use this asterisk because I know that all my user entered keyword information will always be found here in the group with asterisks. So here is my stim condition keyword. I'm just going to select that single one for this example because that's the only condition change. All right, so I've got those settings. I'm going to concatenate. It writes the file to disk. I will bring it back in to my new workspace and close this. So where does it come? It comes back in. Oh, a new workspace. What did I do? Sorry, I meant to open it in my existing workspace, and that's just a checkbox. Here, let's just do it again. I'm going to overwrite the file. Yeah, sure. And then open it in my new workspace instead of opening it in a separate one. I'm sorry, do the same thing. Existing workspace. I just wanted to come back in here. There we go. So I can close that other one. Don't matter. Cancel this and close that workspace. Not save it. So what I wanted to do is get in my new sample, which is right here, concat, right? Lymphocytes. I can put it in my gating group and it'll get all the gates, and it should have 100% of the events down to the population that I used for the concatenation, right? Because all these cells should be included in that gating tree. Now, this is the sample that I'm going to do all of my high parameter dimensionality reduction and clustering analysis on. And we can start just by running a t-SNE. So let's go ahead and use the t-SNE tool right here under the workspace tab, populations band. And you want to pick the compensated parameters minus anything that you may have already gated apart that you aren't, or that are not relevant to your, lineage and such. I'm just going to deselect the live dead, which I already used at the root sample to gate away dead cells. These are all the options. I'm not going to go through them. Know that in most cases you can just push run, and that's the right thing to do. If you start changing these, the only reason I would change KNN and gradient algorithm, or if I'm working with, say, 1 million events or more, and that will speed up the algorithm if you have high numbers of events. So I'm going to click run. It's going to give me a bar showing what it's doing. It's computing the k nearest neighbors, and then it's going to start doing this iterative calculation to embed the data into 2 dimensions. And I have somebody asking, do you think we spend the same amount of time using FlowJo or doing the same analysis on R? So this was a question from somebody just earlier. I'm just going to answer it while this is calculating. I think that it all depends on your coding experience and familiarity with the R statistical computing environment. There are some great tools out there available in R, and it's worthwhile getting to know R. What we've tried to do with the plugins is eliminate the need to code in that language, which is a barrier for a lot of bench scientists and immunologists that just don't have coding language training. And it would take them maybe months to get to the point of using these tools in R that we can provide them just as a point and click interface. In terms of how fast things will run, well, if it's an R-based plugin like FlowSum, it's going to run the same amount of time on the same amount of events, same number of events and parameters. It's going to take the same amount of time to run it in R as it is to run it in FlowJo, calling out to R. In terms of t-SNE, R t-SNE is way faster than the R implementation. And so I would not run t-SNE in R. It just depends on the algorithm and the implementation, right? So R is contributed to by the authors of these algorithms, and these tools are available as packages. I think the real barrier there is really getting familiar with the coding language and being able to operate within it. If you can do that, then all we're doing when we run this is exporting this file, delivering it to R, and then returning back results, right? And in this case, t-SNE is just running natively, and we've optimized it to run way faster than the R implementation. So t-SNE is better here. In terms of some of the other ones, like embed sum or flow sum, well, those are R-based packages anyway. And so it's going to be easier for a new user who's coming into high parameter data and does not have programming skills to use FlowJo and call out to those operations from within the FlowJo UI, even though there's some setup involved initially. It's going to be much challenging, more challenging for somebody. But you know what? I'll give you a suggestion here of where to go if anybody wants to learn R. I mean, R is an awesome, let's see, R for data science. Oops, that's not what I wanted. R for data science. All right, here's what you do. If you want to learn R, you go to this R for data science book, and you start walking through the table of contents. It'll teach you how to use visualizations. It'll teach you how to wrangle data. It'll even teach you how to basically program. And if you work through this set of this book step by step with the examples here, you're suddenly going to come out at the end knowledgeable about this programming environment and probably be able to use it. So it all takes time, right? And it just depends on where you are in your career and whether you focused your time on learning that. So appreciate the question. Now I'm done with my map here. It's got the resolution, and now I want to do something with it. We could also run UMAP on this. We could run clustering on it. So let's go ahead and run a clustering algorithm. I'm just going to use phenotypes because I like phenotypes. Hell, why not use the one that you really like? So phenotypes, you're really just prompted to choose the parameters that you want to consider and set a K value. This is the phenotypes version 2.4. It's running in R. I keep it around in my FlowJo because I'm usually trying to run not just 50,000 events, but hundreds of thousands or even millions of events. And I found that the version 3 that's out, that we have out, doesn't scale quite as well. And I can end up waiting way too long for my clusters using that version. So I just leave the defaults. I like to check this box to save the R script in case there's any error that comes back out, then we know how to troubleshoot it. And I'll just click go. So this is what's going on, right? Export the file into essentially a extra folder that it gets created. It's called the plugin derivatives folder. It delivers that to R, and then it returns back the clustering results. It's going to say calculating while it's doing that process, and it's not going to be immediate. You're going to have to take 30 seconds, maybe 2 minutes to do the clustering on it. So I'll let that run. While it's running, I can still interact with this data. So remember I said we could do a color axis, right? And choose a marker of interest. Where's my interferon gamma cells? Then we could make a gate here around that region, ask what it is, question mark, what is that subset? I'm going to go ahead and right click here, copy to the layout editor, my population here. And then my layout is popping onto my other screen. I apologize for that. So here's my layout. And I'm going to make an overlay by taking that question mark and dropping it on, okay? You can change colors of the overlay. I'm going to take that overlaid subpopulation, make it magenta because it's just my preference, and make it gray on the bottom. And then we can right click, make multi-graphs, histograms. And there's a lot of parameters here, so I might need to go and right click again and choose which parameters I want to include here, and then make that accessory set of plots larger so we can see what's going on. So I'm choosing my parameters, grabbing this one and making it big, right? So what do we got here? We've got a population of cells. It's CD28 positive, interferon gamma, it's a 3, CD4 positive, CCR7 intermediate, right? this population, if I just gated for CCR7, I'd cut right through that. And now my phenograph calculation is done, so it's returning there. Oh, it's CD69, so it's an activated TNF alpha, 127, RO positive, 154 positive, IL-2 presenting, right? It's a polyfunctional IL-2, CD69, TNF, interferon producing TH1 of some sort, right? It's a CD4. So there we go. We got the phenotype. Now, I like that we have these linked. So, you know, we can go over to this population and be like, well, what's that island? Well, that's a CCR7, CD4, CD3, CD28 population, but it's negative for RO and it's rocket high for CD27. And so, you know, we have a different subset and it positions itself differently within the dimensionally reduced data space. So now I've got all these clusters. What do I do with them, right? Well, I'm not going to show you the export because essentially you just, if you wanted to do this with the table editor, you have to take these populations, right click, export them as individual files, make a group, and then enumerate the statistics with your add statistics button for medians of all of these parameters, right? And make a table. Well, now I can just do that using the table or the Cluster Explorer tool. So I'm going to go to my plugins. I'm going to go to Cluster Explorer. And it's going to ask me, well, which groups do I want to look at in terms of cluster populations? What visualization do I want to see them in terms of dimensionally reduced embeddings? And then uncheck the live dead that I don't want to see, click OK. And it's going to pop all these windows up. I'm afraid, yeah, it did. I should have pulled my exterior monitor. So I was popping them all up on my other monitor. I'll have to move over here. So now I've got this, the line plot representing the data. I've got my clusters and all the phenotypes that they represent as a heat map. I also have a control panel here that I can choose these different options from. And I've got 2D plots. The one I spun up was just my T-SNE. So you can see the position or location within T-SNE space for each of your clusters. And this is all linked together. The first thing you probably want those to know that you can click this menu button. Oh, I hate that it does this only when I'm presenting in another one. So yeah, you can choose to copy content, copy the image, or add nodes as columns. So if you copy the content out, these are just, in this case, the means scale values. I could plot medians instead by checking the radio button here. You can gate within this line plot using and not gate. So if I want to use and, maybe I want to look at everything that is CD3 positive. Let's see. I actually don't use this as a demo very often. So CD3 positive, CD4 positive cells, right, that are not FOXP3, not FOXP3 positive. Well, there we go. So here's my populations that fit that definition, right, that I just gated on here in the line plot. I can actually play through these clusters if I want. Right, seeing where they live and what the expression pattern is, pause it. Any of these can be copied as images, right, in the state they're at. And then ultimately, yeah, I'm over time, aren't I? Sorry, guys. We can actually choose different cluster sets here. We can even choose different nodes. So yeah, that's always, whenever I'm displaying this on a screen share, it does that. So I can reset it just by double clicking on a white space anywhere I want, and it'll come back to all of the levels. And again, if I want to, I can actually copy the image here, and I can go paste that image right into my layout editor or into my PowerPoint. All right, oh, interesting, I got the highlighting there. I could close that. I can always close these windows and reopen them, right? Reset it. And then, as I said, you can actually get the frequency numbers out here by going to your menu and choosing the last option, which is select and add nodes as columns. And when I do that, see, I hate this. I'm sorry, I'm now seeing all this red. I've never seen that except for this screen share. I could actually choose my different condition gates there and show them as additional columns, as I mentioned before. Of course, my condition gates don't exist until I create them. So I'm going to close this Cluster Explorer right now, and I'm going to just make some condition gates. My stim condition keyword, this is where I can pull apart my samples. I also always get a sample ID keyword, but sample ID is in the order of the list when you concatenate it. This stim is me saying I want these events to be at 1 and these events to be at 2, so I control where the placement and the order. And so I'm going to call this no stim. and stimulated. And just remember that you can always, you've classified, every cluster has a classification here into, well, sorry, it contains events from stem and no stem, and you can break it apart. So let's go to our layout once more. If we take the root file, let's see if I can get it in here. There we go. No, maybe not. Should have pulled my extra monitor. I'm sorry. There we go. It's going to be on there sometime. Okay. So let's take each of those, the root file here, right? And actually, I'll show you this one. So I'm going to go Tsne 1, Tsne 2 as a top plot, or as a pseudo color top plot. Is just going back to my Tsne parameters, right? So that's everything in the system that is all cells, stem and no stem. So I'm going to duplicate this. Holding down the Option button and dragging will duplicate, or you can right click and duplicate like that. And if I duplicate this twice, then what I'm going to do is over here in the gating hierarchy, I'm actually going to drag and drop, replacing this everything event plot with just the no stem events. And to do this, I'm dragging over, like I was going to make an overlay, but before I drop it, I need to press the Option button and see the little plus button show up there on my cursor. Now, when I drop it, I'm replacing what's in that plot with the selected population. I can do the same thing here with my stem. I realized I did no stem first. That's the way I always like to think things, no stem, and then what happens when you stem. So I'll grab this. I'm dragging without Option first, holding down Option and dropping, and it replaces. So you can see, that there are cells that are present and cells that are gone in the stimulation, such as this query gated population. If I take those two populations, no stem and stem, highlighting them both and drag them to my query gate, well, wherever I place that query gate, this is going to tell me the frequency of that gated population across that's coming from my different conditions, right? And you can see where my gate is on this set of CD4 cells that they're present in the no stem, they're gone practically in the stem, which means that this population gained effector functions, moved to a different location, and was lost upon the stimulation condition. So that's kind of where we're going with this type of high parameter analysis. You need to, again, concatenate and merge those samples together so you have a way to classify all events with the clustering approach and position all events within the same dimensionally reduced embedding. Then you can actually gate apart the different conditions and compare them visually with the dimensionality reduction space, or numerically, statistically, by taking what you put in and seeing how the output comes out in terms of what clusters and how they're distributed across your experiment. Okay, so I went over a little bit. I have a tendency to be long-winded. I hope this was helpful. Should get this recording up on the webinars, previously recorded webinars page in a couple days or a week or so it'll be available to you. If you have any questions, I see one coming across, please go ahead and type them into the Q&A box if you can. Relative expression transformed. How is the signal transformed? Okay, so when she's asking specifically about relative expression, and what I mean by transforming is that this is actually, that visualization is binning the data. So let's spin it up once more, just to see that. It is all relative. All expression levels are relative because it depends on the fluorochrome you use and how bright that is in terms of the stain index. So one HLA-DR antibody is going to have a totally different numbers underlying it for what's high and what's low, depending on its conjugation to a fluorochrome. So this says relative expression level transformed. And this basically means that we're using binned data here from zero, each expression pattern. If we look, let's look at a 2D plot. So I'm just clicking 2D plot. I can make as many of these as I want. There's actually a bunch of options for 2D plots here. I hate that drop down being red like that. I'm sorry. It doesn't happen when I do that on my regular screen. Now see the data space, right? We've got a bi-X transform, we've got different log scales. We basically take the visual data space and we bin it from 0 to 4096. And that is what we represent here as bin data. So scaled or transformed data. We're looking at the transform space. These will change depending on whether you go back to your T button for a parameter and make a change to the axis, right? Customizing the axis here allows you to change the width basis, the positive decades function, the ranges, and then what we're doing is taking that transform data space and chopping it into 4096 bins, and that's what we're visualizing here. So it's all relative expression levels, but in terms of what you get out here, if you actually copy the content, that's going to be controlled by by whether you're plotting medians or means, and whether you're using scale values, which is actually not bin data. So the raw values here, like you were going to get if you applied a median statistic to a population within FlowJo, or channel values, which is binning into 4096 bins. So if I copy the content of channel values here as medians, let's say, then I'm going to get the median of the binned data, and all of the values underlying this heat map are now in integers from 0 to 4095, truthfully. So it's all going to be positive information. When you go into scale values, you're talking about raw data as it's displayed here in the visual. And now you've got actually higher in terms of what numbers you get out. There's the potential in certain colors, of course, that you would get negative, like less than 0 MFIs. But you're looking at that same, in this case, the range of the instrument here was, 262144 is the traditional range on a Fortessa or whatever instrument that you're collecting this on. So I hope that's helpful. Yeah, it's all relative in Flow, right? Depending on the tools you're using. Any other questions before I call it for the day? Well, then in that case, thank you all for your time and attention. We do appreciate you joining us for these webinars. And if you have any questions, don't hesitate to reach out to me. I'm Timothy Crawford at bd.com, or my tech support group, that's just flowjo@bd.com, with questions that you may have in the future. Thanks for your time. Good luck with your research and your analyses. Thank you.